Thanks, Adam. Thanks to the organizers for inviting me to this important event. And I think it's very important today that we start to more intensively communicate with the regulatory bodies, with the decision makers in healthcare and also with industry. And I hope I can contribute a little bit with my outlook into the future of molecular imaging. I go into the concept. I want to underline that we have, if you look back during the last decade, we have a very rapid evolution of technologies, particularly in the area of molecular diagnostics, which is mostly driven by inventions of molecular biology. And if you look here where we are coming from, a mainly morphologically driven diagnostics of tissue slices coming from biopsy samples or primary tumors, which are then stained with antibodies to visualize specific targets which are prognostic or treatment relevant. We went more and more into sophisticated genetic analysis of tumors, a concept which we're calling also omics technologies. So we have different levels of analyzing methods, starting from genetics to reaching to the protein level, proteomics, and many disciplines in between. And I just did a photo from a letterbox of our Center of Molecular Medicine in Vienna, and you can't read it, but there is written overnight DNA sequencing, fast on-site, and green service. So these technologies of sophisticated, detailed genetic evaluations of tissue is available, and not only for research, but also for clinical routine diagnostic. And at the same time, we're recognizing that these kind of technologies are not only available and increasingly done, but it's also very cheap. This slide, because concepts of personalized medicine were introduced nearly a decade ago. This is a publication of Roy Herbst and colleagues published in the New England Journal in 2008. And at this time, they suggested to add to the classical immunohistochemistry analysis, which I showed before, novel technologies of proteomics on the protein level, but also all these sophisticated analysis of underlying gene patterns, so mutation status, deletions, amplifications, and so on, methylation status, so epigenetics. But even at that time, they suggested to also implement blood-based profiling, so a technology which we are calling today liquid biopsy, where you potentially are able to analyzing from blood or also other liquid samples, saliva or urine, you analyze circulating tumor cells or other kinds of circulating fragments. And bringing all this together should then increase the sensitivity of a tumor to a certain treatment. Discussed this before, and it is an ongoing discussion how much we need this, or we need to provide evidence both for treatment, in particular also for our diagnostics. But if you review the clinical trials done in oncology, you will find that if you go for biomarker rationales, so a personalized approach, then you get a higher success rate as compared to the conventional approach. But if you go for new drug approvals in oncology, you end up with around 25% of success rate, which means vice versa, that 3/4 of the trials are failing. And you can imagine what that means, and we heard something about cost effectiveness, what that means for resources and costs in the healthcare system. So we need here strategies to overcome this general limitation. The problem is the challenge of tumor heterogeneity. And what does this mean? We know that tumors are spatially heterogeneous, so there is heterogeneity between driver gene mutations within the tumor, but there is also a inter-lesion heterogeneity. So metastases from a primary, for example, in the lung tissue or the brain tissue must not necessarily be the same as in the primary tissue. This is what we know today, and there is an amount of literature showing exactly that. And what comes also more and more up now is that we also have a temporal heterogeneity. So the tumor biology is changing with every cycle of treatment we are applying. So if you come from a primary tumor or a biopsy at baseline and you perform treatment, you have the situation that the biology with a high probability has changed, and you cannot rely on what you have had at the time point of the first biopsy. So this is temporal heterogeneity. Always argue that, yeah, you have nice diagnostics available, but what does this help if you don't see the tumor? And this is a strong argument we have, and this is the reason why we are doing imaging and showing pictures and all that. I'm not sure if this is a very strong argument for the future, but still we should use this. So the question is, how could nuclear medicine and molecular imaging contribute to these modern concepts which are coming rapidly, which are partly already implemented in the routine diagnostics? Or that the evolution of technologies in the molecular diagnostic field was nicely paralleled by an evolution of the imaging technologies. We heard that. So the field is moving more and more, coming from more morphologically oriented imaging technologies, CT, MRI, into these kind of molecular imaging strategies. And we are able, by the use of radiopharmaceuticals, to characterize the tumors in vivo, also the microenvironment of the tumor. Our argumentation is that we are providing whole body imaging. And you see one of these very nice PET-CT images here, where we can really analyze multiple lesions in the whole body, different tumor regions, because we have the whole tumor. We can do this in real time, serially, non-invasively. So this is really a strong strategy we have available. And by the use of different kinds of radiopharmaceuticals, we can indeed visualize specific treatment-relevant targets. This is what we always have to take in mind. And if you look back the last 10, 15 years, we learned also that tumor biology measured in vivo frequently differs from tumor biology measured ex vivo on tissue slices. You have many problems with standardization. You have different ischemia times until the slicing is performed and all that. So also these, let's say, reference standards of pathology have their limitations, and we have to consider that. You could call this also not necessarily nuclear medicine or molecular imaging. You could call that also in vivo tumor pathology. That because we can use the same drugs or particles, vehicle molecules that bind very specifically with high affinity to the treatment-relevant target, and just by linking these molecules with a radionuclide, which enables then the imaging or treatment, as you heard before, PSMA is such an example. And we also have to recognize that we are dealing with amounts of drugs that are in the picomolar range, and we are gaining a very high molecular resolution in the sub-millimeter area. So we are only limited, let's say, and Jim will speak about that probably in the next presentation, by the scanner technology, which provides resolutions which are higher than the potential molecular resolution. So what is coming more and more is, I call that here, targeted imaging techniques. So we have many of these techniques potentially available. There are other examples. I just show one on breast cancer. Estrogen receptors can be visualized and quantified by a compound called fluoroestradiol. And there is an amount of publication out, and you see a typical example here, metabolically active tumor lesions, mediastinal lymph nodes, which are at the same time FES positive, and here we have metabolically active lymph nodes which are FES negative, very similar to what I showed before for the EGF receptor. And we know, and this is a meta-analysis from 2013, that those patients who show no uptake of the tracer have no receptors and will not respond to treatment. So the negative predictive value is very high, and you can avoid unnecessary treatment and cost-intensive treatment with these kind of strategies. Of course, molecular imaging, and we heard about that also in the previous presentation, has some limitations. If you go for subcellular structures or cellular structures on a single cell level, it's hard, of course, to visualize these in an in vivo setting. We can't do that, so we need somehow a combination with molecular diagnostics. And the clonal evolution, the resistance of clones is hardly detectable with this kind of imaging technologies. So for that, we could combine this with liquid biopsy, for example. So there is a rationale for combining molecular imaging with molecular diagnostics in the future. So this is my summary. I think nuclear medicine has a very high potential to link target identification with treatment and thus to personalized treatment. I think we have a high potential for in vivo tissue characterization to improve biopsy guiding, response prediction, therapy monitoring. And the future vision I showed you is to provide an in vivo characterization of the tumor biology repetitively. We could call this in vivo pathology, virtual biopsy, I don't know what else. And I think there is a complementary use of combining molecular diagnostics and molecular imaging. I think we have to fulfill important tasks, and this will be a matter of discussion later probably. I think we have to increase the availability of molecular whole body imaging techniques and expertise. We have to facilitate the legal aspects for the use of radiopharmaceuticals, make them available, and we have to find ways to finance clinical trials showing the evidence for using these kind of molecular diagnostic strategies.